Okay, no, it should be those.
Okay, cool. It's recording. Let me go to share screen. Okay, can everybody see what I'm looking at just before I get started?
Yeah, the study guide.
Okay, sweet. So we're just gonna work down this. I'm going to basically highlight a region, like highlight 1.1, and then go off to the side and work through 1.1 and kind of get everybody up to speed on everything. Don't let us move forward unless you guys are fully understanding what we just covered. We can take our time and kind of just move through this slow until everybody understands. All right, cool. So we'll just start off the top. Go. Okay, cool. So the first thing we're going to work on is kinetic potential energy and bonds. So first we'll just talk about energy. And energy is basically the capacity to do work. And like always, if you can't read my handwriting, just let me know. Okay, so there's two types of energy. There is, let me see if I can do this. One second, I'm going to change my font size. Cool, there's kinetic energy. So think of something like a moving car. One second. There we go, cool. Sorry about that. So there's kinetic energy. And this is 1/2 mv squared. And there's potential energy. So for potential energy, think of a roller coaster that's about to drop. And there's a lot of potential energy.
We in, folks.
Wait, I want to see too.
Just oh my god, that's how you want me to see.
Your guys' mic is on. So potential energy, you're at the top of this peak, and it's basically energy held in the system that is potential. And then once this would go down, that'd be kinetic. So to relate this to bonds, think of the potential energy being the energy holding that bond together. And then when that bond breaks, let's say if we have propane, there's a lot of potential energy in propane. And when we ignite propane, let's say you're sparking up the grill or something, that propane ignites and it combusts. That's its kinetic energy. The combustion portion is the kinetic energy. So the relationship between electronegativity, bond length, and potential energy is basically, we'll do 2 bonds here. And I'm exaggerating this just to show the point. This is much more electronegative than this one. So the bond length is going to be shorter. So this is kind of a way to memorize this. As electronegativity goes up, this bond length is going to get a lot shorter. And that shorter bond length is going to it's going to be a lot more stable, and there's going to be less potential energy. And if you think about something that's very stable, it's a lot easier to hold together or hold it close than something that's not very stable. So there's more energy required to hold this long bond together that's not as stable. So there's more potential energy, which means it's going to be much more reactive, especially, what the heck? Especially when it's ignited, that bond is going to burst, which is why your carbo carbon chains are a lot more reactive than non-carbon chains. Think of water, and then think of like methane, ethane, propane, all of these explosive gases. And then the reverse is true. As electronegativity goes down, bond length goes up, it's less stable, and it has more potential energy. What are the first and second laws? So for the first law, I like to think total energy in equals total energy out. And then so all spontaneous reactions lead to an increase in entropy. And we know that entropy is the disordering. It's basically the disordering of things. So if you think of this block, and then we have this over here. So this block is very ordered. It's going to have a lot more, it's going to have more potential energy. This one is more disordered. It's going to have less potential energy. This one's going to have more entropy. This one has less entropy. And we can just connect this to what's coming up. This would be an example of catabolism.
Yeah, catabolism, but anabolism for that one.
Yep, so this is catabolism, which is basically the breaking down of something. And I'll do an example that I think is pretty good here in a little bit, but we're not quite there yet. So does everybody have this on their screen? I'm going to erase some of it. Cool. Okay, so now we're going down the Gibbs free energy.
Wait, I have a question, sorry. So if there's like high order, then there's more potential energy and less entropy?
Yep, so I'll draw a picture. So we'll say this is our high order molecule or whatever you want to call it. Okay, so there's a whole lot of bonds going on here, and each of these bonds has some potential energy. And that makes us very stable. But if this thing starts getting broken up, the disorder increased a ton. So the entropy increased, but the enthalpy, which is basically
Heat.
the enthalpy, which is like the energy in the bonds, it decreased a lot, because where this one we had all of these, we had 3 bonds connected to it. Well, now we just have 1 bond holding 2 of them together. So the enthalpy decreased quite a bit. Does that kind of answer that in terms of energy?
So enthalpy decreases when there are less bonds? Or when there are...
Did you say enthalpy?
Yeah, enthalpy?
Yeah, so enthalpy increases when there's less bonds. And don't think of it as more or less bonds. Think of it as energy in the bonds.
So electronegativity, right? So like an HF would be a really electronegative molecule. So would that have more enthalpy or less enthalpy?
This would have less enthalpy than this.
Okay. It would have less because it's more electronegative.
Yep, so it has less because it's electronegative, and the bond length, so I'm just going to do BL's bond length is shorter. So the bond length is shorter, which means it's a lot more stable, and so it has less enthalpy, and it also has potential energy. And saying enthalpy and potential energy, they're interchangeable. It's kind of synonymous, same thing, basically. This one has more potential energy.
Did you erase it? What was the second law of thermodynamics again? How did you phrase it? It was
So I like to phrase the second law of thermodynamics as everything, all spontaneous reactions result in an increase in entropy.
So for the HF, would that be stable or not stable?
It's very stable.
It's very stable.
Yeah, it's extremely stable. Those are really strong hydrogen bonds with that fluorine. So fluorine's very electronegative. It's actually it's the most electronegative atom there is, which and that's a function of its Zeph, which is the effective nuclear charge. So fluorine's a very small molecule, and because it's so small, the protons are very close to that outer shell. So it's going to be able to form relationships with other atoms' electrons very easily because those protons are so close to the outer shell, its valence electrons. And that's why it holds hydrogen so tight to it. Cool. Is everybody good to move on to 1.2? Cool. So going over Gibbs free energy, this is basically an equation that helps us determine if things happen spontaneously or not, and at what temperature. So when I was taking biochem, I kind of just made this little table that I feel like helps me the most. So right off the bat, we'll just talk about G. A negative change of G is spontaneous. A positive change of G is non-spontaneous. And for now on, I'm just going to write spawn and non-spawn because writing out spontaneous kind of sucks. So we have some variables here. We have change of H, change of S, and T. So here's the table I made, and I think it works really good. You have change your enthalpy, your entropy, and then we'll have spontaneous question mark. So first, I go plus plus, minus minus, and I like to make tables really simple and brainless like this because when I take a test right off the bat, I just like to dump all my tables onto the front page. So the way I think about this is you have positive, positive. Think of like a number line. Positive, positive, that's a high number. This is spontaneous at high temperatures. Boom, it's easy. Okay, so minus, minus, same thing. Think of a number line. That's a very low number. This is going to be spontaneous at low temp. All right, does everybody understand those two and why that is? If not, we can throw in some numbers and actually solve for G. Okay, so this next one. So these two are easy to mix up because you can't just think of a number line or like some cheat code. So you have to think. Entropy is decreasing. And usually everything spontaneous is an increase in entropy. So this is going to be non-spawn. And I'm just going to put non-spontaneous, and that's for all temperatures. You can get into some high levels of chemistry and stuff where that doesn't apply. Obviously, there's extremes to any situation where you can make something happen. Like if we heat this thing up and use all the correct enzymes and stuff, like it'll be spontaneous. And then for this one, entropy is increasing. So this is going to be spontaneous. So this is a really good table to memorize. It's easy to just throw down on the front page of your test, and it'll probably help you guys with some problems. So now we'll go through and we will define entropy and enthalpy. I'm going to type it up here. So entropy is disorder in a system or universe. Enthalpy is energy within bonds. So does everybody have this table? So we're going to do a couple examples, and you guys just tell me what happens with enthalpy and entropy. Actually, we can just do one. Okay, so just anybody. For these top ones right here, what is the enthalpy and entropy?
The entropy for the top, when it's 3 squares together going into separate,
Yep.
the entropy is positive, but the enthalpy is negative.
Exactly. So then based off that table we filled out, would this be spontaneous, non-spontaneous?
Top the table.
What was that? Hello? Okay, I don't know who answered that, but this would be spontaneous.
So the delta H, is that the change in enthalpy?
Yep, so that's your enthalpy, energy within the bonds. So if you look at here,
So
there's,
it's not like heat. For some reason, I see the delta H and I think temperature, I think heat.
yeah, so you could think heat because it's a potential energy, which energy is, heat is usually energy. It's a form of energy, usually
I
kinetic.
thought it required energy to break bonds.
Yep, so it does require energy to break bonds, but the degradation of this is going to be spontaneous. But you're right, it does require energy to break bonds. And
So
making
wouldn't delta H be positive?
nope, because we're not looking. Okay, so for your delta H, this is energy within the bonds. So these aren't making bonds with each other. We went from, think of these bonds right here, 1, 2, 3 bonds to 0 bonds. So this one had much higher potential energy. If you want to think of this, you could think of it as, let's think of it as a carbon structure. And then it's getting burst into individual carbons. So think of propane. We ignite that propane. The ignition is very spontaneous. You get to cook your food, all that stuff. It makes this hot flame. There's tons of energy that's given off. And then you're left with these carbons that float up into the atmosphere. Does that kind of put it into a real world example?
I thought that, so aren't you breaking bonds here?
Yep, so the bonds were broken.
Right, so wouldn't that require so why wouldn't it require an input of energy?
Well, think of, let's use the same propane example. What is the energy used when you light that propane?
You need fire to light it.
So think if your entire living room was filled with propane and you lit a lighter, do you need to go and light each individual propane molecule? Or when you light one, is it just spontaneously all explode?
I guess you would need one, but that's still an input of energy.
Yep. And you got to think everything requires an input of energy. So think of your energy graph. Everything has this little input of energy that's required. You'll never see a reaction graph that goes like that.
Mm-hmm.
Everything requires this little bit. It's called the activation energy. Everything
Right.
requires the activation energy.
I think
So this would release energy then?
Yep, there'd be a big release of energy.
wouldn't that be relative to just the activation energy, right?
The heat that's put out?
Yeah, so I guess she's kind of asking like it requires energy to get the reaction started, but the overall energy that's released is much greater.
Yeah, exactly. And the energy that's released is so great, it'll just basically like domino effect.
Did you say it's an exothermic reaction?
This would be exothermic.
All right.
And this is also a form of catabolism. All right, and then for this bottom one, it's going to be the total opposite. So our enthalpy is going to increase because we're forming bonds, and our entropy decreases because we're actually removing disorder. And this is a form of anabolism. So we're forming bonds, so we need energy input. So I'll just go down to, does everybody have this? I'm kind of going to draw the same thing, but just with catabolism and anabolism. Cool. So down here. Okay, so we just mentioned that this is, and this is catabolism. So when I think of this, when I think of anabolism, I like to think about anabolic steroids. And I think of somebody at the gym trying to get really big. They're using anabolic steroids and they're creating this muscle mass. That's kind of how I remember that. Anabolism is small going to big, or you could think of your macromolecules and your monomers. Now, if you're taking anabolic steroids, think of this person that's going to the gym taking anabolic steroids. They also need to take other things like amino acids and protein shakes and weight gain and all this stuff, eat a ton of food. And that's because they need ATP to fuel these reactions. So this guy who's going to the gym, taking his anabolic steroids, he requires a lot of ATP to make all these bonds. He's building new tissue. He's building, increasing his tendons and ligaments. All this stuff is requiring ATP to grow and get bigger. So where does he get it all from? He eats food. He eats macromolecules. So when he eats these macromolecules and they go into his system, they're going to go through a bunch of catabolic reactions, and they're going to get broken down into monomers. So when he eats all this food, he's going to produce ATP. So anabolism and catabolism aren't always paired up like this. I just paired it like that because I think it makes for a good example. But when you think of catabolism, it's going to be paired with ADP to ATP reactions. Think of glycolysis. You get a sugar molecule, and it's getting broken down further and further. And every time it's broken down further and further, it produces some sort of energy carrier, whether it be ATP, NADH, or FADH. And then for anabolism, why do we go from ATP to ADP? It's because we're forming bonds. So here, there's no bonds here between any of these. In order to form a bond, you need to put in energy. It's endothermic, so it requires this is EXA. So it's endothermic, so it requires energy input.
So would the ATP molecule be smaller than the ADP?
ATP is a larger molecule.
So wouldn't it be anabolism from ADP to ATP and then catabolism from ATP to ADP?
Nope. So during anabolism, you require energy. So let's look at here. So we had, I'm just going to do it. That's the adenosine. And then this is our triphosphate. So in order to make this, we'll say we have to make these bonds right here. This phosphate is going to be cleaved off because there's potential energy in this bond. And when this is cleaved off, there's going to be a release of energy, and that energy will be enough to form these bonds.
So ADP contains more energy than ATP.
No, ATP contains more. So this, what this arrow shows is this, So this is ADP, and this is ATP. You have to think these phosphate bonds, that's a bond as well, and there's potential energy within that bond. So when we disrupt one of those phosphate bonds, it's going to release energy, and this energy can be used to fuel an endothermic reaction, like forming a bond. If you guys remember the peptide bonds that form between amino acids, that also requires some sort of energy input. You're going to see a lot of things in biology that use this ATP to ADP reaction, and they take energy from that ATP bond. Over here, catabolism. Think of our glucose steps where we're breaking down. Can everybody see what I'm looking at on the website?
Yeah.
So look at these areas where ATP goes to ADP. So we have an ATP to ADP right here. And then we have an ADP to ATP here. So glucose to glucose 6-phosphate, we were making a bond. There's an extra phosphate that goes in here. So ATP was used, one, to put the phosphate on that bond, and two, it was energy put into the system. Over here, we have a big molecule going to a smaller molecule, so catabolism, and it results in ATP. So the best way to wrap your head around catabolism and anabolism is to use real-world examples. I definitely would not recommend looking at a graph like what I have written here and trying to memorize it all. Memorize this graph for like a data dump at the beginning of the test, but really understand how these concepts apply to pathways and stuff like that, especially with glycolysis, the Krebs cycle, all that.
Hey, Dequan.
Yeah.
There's a question. So exergonic, so like gonic would refer to energy and thermic refers to heat. Is that right?
Oh yeah, that's right.
Okay.
Yeah, so this could be exothermic. Yeah, it's going to give off energy and heat. It's basically synonymous. Energy is heat. It's just kinetic energy is heat. It's the movement of molecules going really fast and stuff. Does everybody feel comfortable with this? Because this can kind of be tricky. Everybody feels comfortable? Okay.
So I'm just wondering, so you don't think of catabolic as like breaking up bonds then, or breaking up a bigger molecule into a smaller molecule?
Yeah, that's exactly what it is.
Right. So why would ADP going to ATP be catabolic?
So when these big bonds are broken down, that potential energy that was in those bonds is released. So think of that potential energy that's in all these bonds, right? And then when that potential energy gets released, it's enough energy to convert an ADP to an ATP. And in some cases, it's enough to make it multiple ATPs.
And that's catabolism?
Yeah, that's catabolism. Things getting broken down into their smaller units. It's exothermic. It decreases potential energy. It increases entropy. It releases heat. It releases energy.
All right. Okay. Thank you.
And and then the reverse as well. In order to get these individual units and put them together, that's going to require energy. Think of if you had a bunch of building blocks and you want to put them together, you have to put energy into that. Or if you wanted to bake a cake, you have your eggs and you have your flour and your water and all this stuff, these individual units. Well, to bake the cake, you got to throw it in the oven and you got to heat the oven up and apply heat energy and stuff like that. But if you just put the cake out on the counter, eventually, I mean, it might take a while, but eventually it's going to spontaneously break down. So there's a ton of examples. If this is pretty tricky for you, definitely check out like a bunch of YouTube videos and just type in catabolism, anabolism, like real world examples or something like that.
Can we assume that all the energy is heat?
I wouldn't assume all the energy is heat. I don't think you guys would be asked a question that in depth where you're having to calculate heat energy and bond creation energy and all this stuff. But yeah, most of the energy given off is heat, especially in something that's a carbon chain. Usually when carbon chains burn up, they're going to have heat.
All right, one more.
Yes.
So for the catabolism and the anabolism, whatever. So you're referring to the macro to the monomers, and then to the monomers to the macro, not the ADP to the ATP, right?
Exactly. Yeah, exactly.
Okay.
Yeah. Good point. Good point, actually. Yeah, you're not the first person to bring that up. I should have mentioned that. So when we're talking about anabolism, we're talking about this red arrow right here. We're not talking about that. Cool. So I'm going to erase this if everybody has it. And we're going to go on to the next thing. So we're going to talk about enzymes. Rachel, nice. Come here. And one more line. Okay, so this is change in energy. This is time. So what we're looking at right now is an energy diagram. And it's basically starting with your reactants, moving down to your products. And what this shows us is the energy of our reactants, the energy of a transition state, which I'll label like that, and then the energy of our products. So if we're moving down this much in energy, that means a lot of energy was given off. We went from high energy to low energy. So think of this
as exothermic.
Yeah, exactly. Think of this as exothermic. Energy is given off, and the products are a much lower energy. So they're going to be a lot less reactive, a lot more stable. And this right here, this gap, that's your activation energy, EA. So what enzymes do is they help lower this activation energy. So we have a blue line and a green line. Which line do you guys think represents the reaction that's happening with an enzyme?
The green one.
Yeah, so the green one lowers the activation energy. So basically, this could be our enzyme. And then let's say these reactions are substrates. And they go in here. And then our product is that. The enzyme is going to facilitate this reaction, and it's going to lower the energy of the transition state. So where we have this, so this would be our circle and square. This would be our circle and square kind of fusing. We'll draw it like that. And then this is the final product. An enzyme is going to lower the energy of this state right here. And that's going to make the energy, the reaction happen at a lower amount of energy, but it's not going to increase the speed. It's not going to shift the equilibrium, and it's not going to change the final energy of the products. All of those are state functions, so they're not going to change with the use of an enzyme. So just make sure you're aware of that on the test, that an enzyme will make the reaction happen at a lower energy level, but it's not going to increase the speed shift the equilibrium or change the energy level of the products, which is why both these energy levels finish around the same area. And also, enzymes are proteins. They're usually a tertiary structure or quaternary structure protein. So things that can affect enzymes like pH, temperature, stuff like that can really mess up a lot of reactions that depend on an enzyme to get past their activation energy. I guess a real world example that's really good to use is think of if you get a viral infection, your body increases temperature in an attempt to denature the viral structure. But it's kind of total warfare because if your temperature gets too high for too long, you're going to start denaturing proteins and enzymes that are crucial to reactions that you depend on to survive, which is why high fevers can lead to death or other nasty things.
Quick question: are we going to discuss the hydrophobic, hydrophilic, and the charged nature of enzymes too?
Yeah, we'll hit that too. So what is an active site? We just talked about the active site. Cool. So I'm going to erase this if everybody has this energy diagram. Also, if there's an energy diagram that looks like this, that's an endothermic reaction, which means there's a bunch of heat that went into the system. So know what your endothermic and exothermic graphs look like. So we're going to talk about active site chemistry, which kind of is similar to last test. So we'll say this is our active site. And we'll just go through these one by one. So we'll say we have a hydrophobic active site. And these are potential substrates that could go in here. Hydrophobic, hydrophilic. So we'll say this is phobic, philic, charged, negative. I should have drawn that smaller. Cool. And then we have, this would be a charge positive. Positive sign. Okay, so if we have a hydrophobic active site, which one of these do you think is going to fit into that active site?
Hydrophobic one.
Yeah, so the hydrophobic one is going to fit in here best. Now, let's say I switch some amino acids on this and I turned it into a hydrophilic. What do you think would happen?
It wouldn't bond.
Yeah, you're not going to go in there. So let's say we want to do a study on a mouse, and this mouse has an active site that we want to experiment with. And we know the structure of the protein that goes into this active site. And we genetically engineer the structure to, instead of being hydrophobic, turn it to hydrophilic. We could create some sort of knockout test and see how the mouse reacts when this is knocked out, this active site is knocked out. So there's a lot of ways you can actually change it from a hydrophobic to hydrophilic, which would not work in there. Now let's say your active site is, we'll say your active site is hydrophilic and it's charged with positives. Which one is going to fit into that area?
The negatively charged ones.
Yep, exactly, because we know it's hydrophilic charged, so we know we have to select from one of the charged, and then we know that opposite charges attract. So what are some negative amino acids? I think lysine is negative. No, sorry, aspartic acid and glutamic acid. So let's say we have a protein that has a high concentration of glutamic acid residues on it, and that's the part that actually interacts with this. So there's a lot of, I think this is glutamic acid. There's glutamic acid residues all over this part. What are some amino acids that we could switch it to that would create a knockout test that would basically shut down this active site, or at least this protein fitting in.
Wouldn't it have to be hydrophilic?
You could do a hydrophilic one. You could do a charged amino acid, a charged positive, and that would create repulsion.
Okay. So
Um,
could negative fit in there and block the active site?
nope. So you wouldn't want to use a negative, unless you're trying to do some sort of competitive inhibition. But if you just want to completely avoid this active site, you're going to want to use a charged positive or something that's phobic. The charged positive would be best because it would exhibit repulsion. Not only would it just not go there, it would repel from it. Okay, so we talked about the active site chemistry and how you're going to look for it. So basically, phobic active site goes to phobic substrate. Philic goes to philic. And then with your charged, just know plus, minus, minus, plus. And know how to block an active site. Or if you're given a question like, let's say there's this active site that is charged negative and you need to build a protein to fit inside of it that has a high affinity for it. And you're given some options of amino acids. Which amino acids would be best on your protein? You're going to want to pick the one that has the amino acids that are the opposite charge. Or if you have a phobic active site, you're going to want to pick the ones that have amino acids that are hydrophobic. Okay, cool. So how does everybody, predict shape. Oh, we just talked about the free energy diagram. Cool. So how does everybody feel about section 1 energetics? Is there anything you want to go brush back up on? Anything that I could explain better? Anything at all? Cool. We'll move on to cellular respiration. If anything pops up in your head, like, oh crap, I forgot something, just let me know and we can go back. It's no biggie. So identify where respiratory reactions are occurring within an animal or plant cell. So I'm going to draw this a little bigger. This is a massive mitochondria. Check me if I messed this up because sometimes I get confused with the pyruvate dehydrogenase complex. So glycolysis, holy cow. So glycolysis is going to happen in the cytoplasm. And glycolysis is the lysing of a glucose. So the glucose comes in, it gets broken down, and I gotta move this. So when the glucose molecule comes down, it'll basically there's 2 stages. So you have the work in stage, and then energy out. So in the work in, your glucose is basically going to go through these reactions, and it requires 2 ATP. This will get broken down further to a molecule called pyruvate. And as it's getting broken down to pyruvate, these phosphates that were added into the system will basically get broken off, and you're going to get 4 ATP out. for a total of 2 ATP. So that's happening in the cytoplasm of the cell. So now that we have these pyruvates, we need to convert this pyruvate to something that we can work with. So there's a phase called pyruvate oxidation, and it's done by the pyruvate dehydrogenase complex. And this happens in the kind of the outside wall of the mitochondrial cell. So pyruvate oxidation. And we'll say this is glucose to pyruvate. Net ATP plus 2. Boom. This is pyruvate to acetyl CoA. So a lot of these reactions will have an output of carbon dioxide, and that is when we breathe out, we're breathing out that carbon dioxide. The carbon dioxide will go into our blood. The blood will filter with our lungs, and through pressure gradients, it'll take in oxygen and release the CO2. So the pyruvate oxidation happens in the mitochondrial cell wall, and it puts in acetyl CoA. This acetyl CoA goes through this thing called the TCA cycle. And the TCA cycle happens in the cytoplasm of the mitochondria. So the cytoplasm of the mitochondria, what the heck? And this gives off a ton of stuff. So the TCA cycle, we'll go take a look at it. It can get a bit confusing. You guys don't necessarily, don't memorize this. Wait till biochem to memorize all this. But just know the main important things. So that's going to be your redox reactions and your energy carriers.
Could you use Dr. Bang's slides? I think those were easier too. They had
Dr.
less. Yeah, for cellular respiration.
Oh, I'll try to bring up one of his slides. Let me see how quickly I can get to it.
So TCA, what exactly was that again? Was that the citric acid cycle?
So it's the citric acid cycle or the tricarboxylic acid cycle. TCA stands for tricarboxylic acid.
Okay, but it's the same process?
Yeah, same thing. It's also called the Krebs cycle.
Where does that occur again?
That occurs in the cytoplasm of the mitochondrial cell. Let's see. Okay, cellular respiration. Yeah, he probably has a simplified version of the Krebs cycle in here. Cool. So we just touched on glucose. Everybody feeling good with this?
Is the mitochondrial cytoplasm, is that the same as the mitochondrial matrix?
Mitochondrial cell. No, sorry. Yeah, I'm pretty sure they're different. I'm not too sure. I think the cytoplasm is the outside and the matrix is the within the other membrane that I drew.
Okay, so it's
Yeah, sorry.
so the big thing, that's the whole cell. The smaller circle is the mitochondria, and that little squiggly thing, that's the mitochondrial matrix inside of that.
Yeah, and if I'm drawing it wrong, definitely check me.
Okay.
So basically, the Krebs cycle, the Krebs cycle is going to pump out all of these NADHs, and these NADHs and FADHs, they carry energy with them, and they're going to go into the electron transport chain. So in the electron transport chain, which is in the actual membrane of the mitochondria, we'll draw it here. So you have these complexes. Whatever, that's fine. So in complex 1, this is your NADH oxidoreductase. So this is where all your NADH is going to go. And when NADH goes through here, it's going to bring a proton down here. Boom, another proton's going to go down. And you have these things called, not cytokines, cytochrome C. And cytochrome C and all those are going to, they basically are like little school buses for the protons in charge. So one second, I'm going to try to go to a, does he have anything on the electron transport chain? I don't think so. So this hydrogen gradient is going to keep building. And it's going to create basically an electrochemical gradient. And this ATP synthase it has 3 parts to it.
I thought
What was that?
NADH went in and then NAD plus went in and NADH came out.
Nope.
Because isn't that a reduction?
Yeah, so NADH is being oxidized.
Oh.
Yeah, NADH gets oxidized, and the hydrogen comes through the membrane, and that's how that hydrogen gradient actually builds up.
Okay, because I could have sworn that.
No, so
Yeah,
think about if it was the other way around, think of what would happen to all those NADHs that are produced by the Krebs cycle.
Oh, I might just be mixing it up or something like that.
Wait, the Krebs cycle was the previous one. Where is the Krebs cycle?
So the Krebs cycle happens within the cytoplasm of the mitochondria.
So like the citric acid cycle is the Krebs cycle?
Yep, it's all the same thing. TCA, citric acid cycle, or the Krebs cycle are all things, they're all the same thing. I don't know why it has so many.
Oh, okay. So there's like glycolysis is first, and then pyruvate oxidation, and then Krebs. And now we're at the electron transport chain.
We have glycolysis, and then this goes down. Pyruvate oxidation. This goes, I'm just going to write TCA, but remember it can be anything. It can be TCA, Krebs cycle, carboxylic acid cycle, and this goes to electron transport chain. So let's say you eat some food. The glycolysis gets broken down. It gets broken down into pyruvate. This pyruvate then gets oxidized via the pyruvate dehydrogenase complex, and it turns into this thing called acetyl-CoA. Acetyl-CoA goes through the TCA cycle, Krebs cycle, citric acid cycle, whatever you want to call it. And when it goes through, it pumps out a ton of NADH and FADH2, and then also some ATP and a GTP. Then all of those NADHs and FADHs go into the electron transport chain. And these complexes use their energy, the potential energy that's in that hydrogen bond, through oxidation reduction reactions to bring in a hydrogen on the other side of this membrane. And when it brings that hydrogen to the other side of the membrane, it creates this electrochemical gradient. Think of like a hydro plant, like a dam or something. And then all of these hydrogens, and they're trying to push up to get back out here, go through ATP synthase. ATP synthase basically has this head that has three different, and it's an open, tight, and closed formation. So when it's open, it's going to bring in a ADP and a phosphorus or a phosphate, and then it'll tighten up. It'll make that reaction happen by squeezing them together, basically, and then it releases them. It's actually a really cool reaction. There's a lot of research in that area right now. And basically humans were so good at endurance sports and things like that because our ATP synthase, our circle has 9 areas for hydrogens, where a lot of other animals have like 12 and 13. So where we can produce, it's like 3 ATP per spin. So with one spin, we can produce 3 ATP. Other animals have to go through a lot more spins to produce the same amount. So our hydrogen to ATP ratio in terms of concentration gradient is way lower than other animals. And if you could genetically engineer something to have like a 6-ring circle or a 5-ring circle, they'd be a complete monster. They've like infinite endurance, basically.
So once the hydrogens go through ATP synthase, then you use the potential energy of that to break down ADP into ATP.
Yeah, exactly. So the potential energy that's created from this buildup of hydrogens is used by ATP synthase. The energy of that gradient is used to convert an ADP to an ATP.
Okay.
Yeah. And then
So that's like breaking it down?
What was that?
Is that breaking ADP down into ATP?
No, so ADP doesn't break down to ATP. It's actually building up to ATP. So this potential energy of the gradient is actually enough to form a bond. Because when you go from ADP to ATP, remember this is, so let me type this out.
Sorry, I thought it was like catabolism, going from ADP to ATP.
So ADP, sorry, I put tri, it's a di. ADP is adenosine diphosphate. ATP is adenosine triphosphate, which means there's an extra phosphate that was added. So where this is ADP, we have diphosphate. This is ATP.
So, but it's still, I thought it was catabolism. That, do you know what I mean?
So if we're talking about this isn't catabolism, this is anabolism, because we're building, we're getting something and making it bigger. So think of the dude eating steroids. ADP going to ATP is anabolic. It's anabolic, it's non-spontaneous. It requires energy.
Wait, what about the previous, sorry, I'm in like the first section, you said that ADP going to ATP was anabolic.
No, I said monomers going to macromolecules is anabolic.
Oh, it doesn't have to do with ATP and ADP?
No, that's just that's your energy source to make the reaction. That's you either need ATP to make the anabolic reaction happen, or the catabolic reaction gives off ATP. Somebody else asked about that as well.
Oh, that's why I was confused.
Independent of the stuff.
Okay. So you use the energy from breaking down ATP you use that energy to break down things?
So when something breaks down, say glucose, it's going to give off ATP. Catabolic reactions give off ATP.
Okay, that makes sense.
Byproduct. Yeah, and then anabolic reactions, they require ATP. So when you think of anabolic, think of we're getting all these little things and we're making them have bonds together. That's going to require some energy. Things don't just naturally form bonds with each other like that. So then catabolism is just the breakdown, exact opposite.
Thank you.
Mm-hmm. And did that explain it good enough? Because sometimes that can be tricky, especially when you draw the graph that has everything in it.
I'm good. Thank you.
Okay, cool. Okay, cool. So we're going to go to 2.2, and we're going to talk about redox reactions. So we will use six. Cool. So here we have H2O going to O2. And then the hydrogen is leaving. So this is oxidation because there's a loss of hydrogens. So I like to remember oil rig. Oxidation is loss. And then reduction, so we have there we go. Going to, holy cow, I'm not even going to draw this molecule. I'm just going to draw that. So reduction, you think of rig.
Wait, isn't that photosynthesis?
Yeah, so this is a photosynthesis reaction, but I'm just explaining a redox right now. We could use any reaction. So reduction is gain. So basically, when you're looking at something like this, if you're shown an equation like this and asked what's being reduced, what's being oxidized, you want to just look at the initial thing. So we have CO2, and then we look over here and we can see that it was gaining hydrogens. On the reverse side, we have this H2O that gets converted to O2. An easier example, Jay, do you know any like one-step redox reactions that are super simple?
One-step redox reactions.
Just where it's like a really basic.
So you did water to oxygen, or oh, that's oxidation.
It's actually kind of tricky. I can't think of any off the top of my head.
I kind of.
Oh, you know what? We'll just look at the.
No,
There's tons on the gas cycle. So every single one of these is a redox reaction. And you can tell what's being reduced because it gains electrons. So all of these are being reduced, which means this is being oxidized. It's always a kind of a two-way thing. When one thing is reducing, the other thing is getting oxidized. So if we go back up here, this is getting oxidized, which means it's the reducing. So this would be the reducing agent. This would be oxidizing agent. So the oxidizing agent oxidizes something and it gets reduced in the process. So reducing agent So oxidizing agents, it will oxidize another species and is reduced in the process. So if you see this oxidizing agent was reduced, and in doing that reduction, it oxidized H2O. Let's see, what are the overall inputs and outputs?
So is NADH a reducing agent?
So NADH would be a reducing agent, yep. And let's look at an example. So we'll look at one of those complex drawings that actually shows the chemical structure. And I think that would be really good in actually seeing the reduction take place. Oh gosh, this is a YouTube video. Okay, cool. So if we look for an area where NADH is, we'll focus on this area down here. NAD was reduced to NADH. So then that means this right here is going through oxidation, which oxidation is loss. You can see where it was oxidized right here. It had the loss as well as here. So anytime you're seeing this right here, this is another FAD was reduced, which means this was oxidized. Same here, same here, here, here. So there's a ton of redox reactions happening. And a lot of times they happen as a coupled reaction to make something that wouldn't be as spontaneous. Okay, so I think that's about it for 1.1 to 2.4. Does anybody have questions on anything at all that we just covered? Okay, if any of you guys do have questions, just shoot me an email or one of the other TAs. There's a ton of people that can help you understand it. And there's also, sometimes not everybody's the best at explaining everything. So if I didn't explain something well, definitely check out like some YouTube videos. Khan Academy is great. Leah for Science is really good. There's a bunch of good things y'all find. And I think Jay is going to take over and go through the rest of the things. Let me switch you to the host so you can.
I don't think you need to do that actually.
Oh, really?
Yeah, let me know if you guys can see my screen. Okay, can you guys see that?
Yeah, I can see it.
Okay. All right, so we stopped off at 2.4. So now 2.5, I'm gonna give a little example of question that could be asked for cellular respiration. Okay, so we learned in class that about 2,4-dinitrophenol, which is known as DNP. So it was a drug that was used around the 1930s for weight loss. And basically what it does is it increases your metabolism and it releases a potential energy that's stored in the hydrogen ion electrochemical gradient. And people basically died of overheating or hypothermia. And because this happens, it will signal other parts of your body to increase cellular respiration and kind of digest food a lot faster because your body isn't getting the ATPs. It's known as an electron transport chain uncoupler because it uncouples the reaction going from ADP to ATP here in the electron transport chain. And the molecule, the drug, is also freely permeable to the inner mitochondrial membrane, which is super important to understand how this works. So we'll just kind of go over it real quick. So this molecule right here will be in the inner membrane space. And since we have a proton gradient here, a high amount of proton gradient in the inner membrane space, it will become protonated and we'll have that hydroxyl group with the hydrogen on it. And instead of those hydrogen ions going through ATP synthase to make ATP, this molecule is actually just going to be freely permeable to the membrane and will go out into the matrix, taking along with it those hydrogen ions. And because this happens, we're not going to be able to produce our ATP right here. So once it gets out into the matrix, it's going to get deprotonated because there's less hydrogen ions there. So it's going to have the negative on that oxygen. And basically, it will go back into the inner membrane space, pick up another hydrogen, and this process will kind of repeat over and over again. And it won't allow for the formation of ATP, which your body needs to do anything. And so basically, this is why people were dying of this drug. So for questions like this, it's important to know kind of the inputs and outputs of each part of cellular respiration and track, like, if there is another question that relates to like glycolysis or citric acid cycle, you guys would know how to do it if you know the inputs and outputs. But this one right here relates to the electron chain. So that's one question on DNP. So does that make sense for everyone?
There's a question to go over that process one more time, just like summarized.
Go over it one more time. Can you guys hear me?
Jessica, you can unmute your mic, and is there a specific part? Just the outputs? Yeah, just the outputs.
Okay. So basically for the electron transport chain, what we get out is ATP, right? That's going to be our output. And for that, we need our hydrogen ions to go through ATP synthase. Okay, so during the electron transport chain, we're going to create this high hydrogen ion concentration gradient in the inner membrane space. Remember, when all those electron carriers are going to be going into those complex 1, complex 2, we're going to make that hydrogen ion concentration gradient. And ultimately, in a normal cell, what happens is those hydrogen ions will go through ATP synthase, and it acts like a molecular motor, and it rotates, and it helps in the formation of ATP. So going from ADP to ATP, right? So we need those hydrogen ions to go through that enzyme. And what this drug does is it basically will protonate a hydrogen right here. And once it protonates that hydrogen, it won't go through ATP synthase anymore, and rather it will go out the membrane into the matrix. So we're not going to have any ATP production. When it gets out into the matrix, it will become deprotonated, and then it'll go back into the inner membrane space, picking up those other hydrogen ions. And this process will repeat over and over again. And basically, that's what ends up killing you, because you're not going to have the energy or the ATP that your body requires. Does that make sense? Okay. All right, so moving on to photosynthesis. We'll talk about where everything occurs first. So the chloroplast is a site of photosynthesis, which is this organelle right here. Light reactions occur in the thylakoid membrane. Let's see this right here. The dark reactions occur in the stroma, which is kind of just like the outside part of the chloroplast. And the ATP synthase that we that's in the thylakoid, or that's used in photosynthesis, is located in the thylakoid membrane. Okay, so if we're comparing the chloroplast and the mitochondria and how the ATP synthases are kind of situated, for the mitochondria, we're going to have this have the hydrogens flow out into the matrix. So we're going to create that ion concentration gradient in the inner membrane space. But for photosynthesis, we're going to create our hydrogen ion concentration gradient in the thylakoid lumen, and it's going to go out into the stroma. All right, so for major inputs and outputs of photosynthesis compared to cellular respiration, we can kind of think of it just as the reverse reactions, both of those. So for cellular respiration, we'll go from glucose and oxygen, making carbon dioxide and water. And photosynthesis, just the reverse of that, right? Cellular respiration, we're going to get ATP and heat. For photosynthesis, we're going to need light energy to start. And one thing to note is plants will be able to do both photosynthesis and cellular respiration. So when we get carbon dioxide from cellular respiration in plants, it can be then used again to go back into the photosynthesis that's needed. And then the other carbon dioxide can come from the atmosphere as well. So essentially in photosynthesis, what we're going to be doing is we're going to be making glucose. So from carbon dioxide to glucose, we're going to be building up a bigger molecule. And so what we learned before is that if we're building up something, it's an anabolic reaction. So we can say that photosynthesis is endergonic. It's the overall process of photosynthesis. And for cellular respiration, since we're going from a bigger molecule down to a smaller one, so from glucose to carbon dioxide, we're breaking it down, or it's a catabolic reaction. So that's going to be exergonic. Okay, so does that make sense? Can you guys hear me?
I can hear
Yeah, I
you.
can hear you.
Okay, can I move on? Okay.
Yeah.
All right, so we'll talk about the light reactions, and then we'll go and talk about the dark reactions after that. So the light reactions will require light energy. It happens in the thylakoid membrane, which contain photosystems. And photosystems are basically like these complexes that will take in the light energy in a pigment known as chlorophyll, which are these smaller green pigments right here. And that light energy will ultimately excite an electron. And once that electron excited, it will follow a pattern known as a Z-scheme, which flows between photosystem 2 and photosystem 1. And we'll look at that in more detail right here. So P680 is photosystem 2. P700 is photosystem 1. And one thing I want to mention is it's called a Z-scheme because it kind of looks like it's like a sideways Z. So a helpful way of remembering this is if you just draw like a Z on your paper for the exam, and you label each parts of the each points of the Z, and remember what happens in each part of that, it will be really helpful to kind of remember how, or remember the inputs and outputs of this. So let's see here. So what happens is we have our light energy. It's going to hit our photosystem 2. That's going to excite our electron, and it's going to flow down a series of like electron carrying molecules like cytochrome, B6, F complex, and plastocyanin. But I don't think you guys need to know that in too much detail. And so once it goes down there, it ultimately goes to photosystem 1, where we have light energy again. It's going to excite that electron in photosystem 1, and ultimately the electrons are going to go to NADPH. So once we lose this electron in P680, we're going to need something to replace it with, and that's where water comes in. Water, when P680's electrons are lost to P700, it's going to be oxidized, and it's going to become a really good oxidizing agent, and it's going to ultimately take the electrons from water, and the byproduct we get out of water is oxygen. So that's where the oxygen comes from in plants. So we can take a closer look at this right here. So we have our light energy. It's going to strike photosystem 1, or photosystem 2, sorry. It's going to be excited and go to BP680 star, meaning it's just excited electrons. And ultimately, the electrons are going to be lost to photosystem 1. And then it's going to go into P680 plus, meaning that it's oxidized, meaning that it loses that electron. So ultimately, it needs to get that electron back again. And this electron comes from water, and it goes back into P680, and then the cycle just kind of continues like that. Okay. Does that make sense? Anyone have any questions on that? Okay, let's move on then. So during all of that, this occurs in the thylakoid membrane, like I said. Okay, you want me to explain the cycle again? Sure. So we have P680, which is photosystem 2. What happens first is our light, the photons of light, are going to come and hit photosystem 2. So that's kind of the kinetic energy that's going into photosynthesis. It's going to hit photosystem 2, and it's going to end up exciting those electrons, and it's going to go into P680 star, meaning that it's just excited. So it's going to go from P680 to a higher energy state of those electrons. And then those electrons are gonna go to photosystem 1. So they're gonna travel down all these complexes right here, and they're gonna end up at photosystem 1. Once the electron gets to photosystem 1, it will become excited again, and it will ultimately go to NADPH. But if we're looking at just photosystem 2, and where it gets those extra electrons from is ultimately from water. So when these electrons are lost to photosystem 1, it needs to gain those electrons from somewhere else. So where it gets it from is water. So photosystem 2 will oxidize, will oxidize water and get the electrons from there. I'll go down to this other diagram here that will help explain it a little more. So again, we have our light energy. It's going to excite photosystem 2, so P680 right here. It's going to excite photosystem 2. The electrons that are excited are going to be kind of just passed down like this electron transport chain. It's going to go through all these complexes, cytochrome B6F, plastocyan, and ultimately it's going to reach photosystem 2, or photosystem 1, sorry. Once it reaches photosystem 1, we're going to have light energy come, and it's going to strike those electrons again. And ultimately, those electrons are going to end up and they're going to go to NADPH. And all while this process is happening, just like the electron transport chain in cellular respiration in the mitochondria, we're going to be creating our hydrogen ion gradient as well, right? So, and we're going to also have oxygen as a byproduct. So once we get those hydrogen ions, this is going to be the kind of similar process where they're going to go through this enzyme ATP synthase, and we're going to make ATP. Okay, so we can say the major outputs of the light reactions are going to be NADPH and ATP. So those NADPH and ATP that we make in the light reactions are going to be used in the Calvin cycle. Okay.
The P680 gets reduced or oxidized?
What was that?
The P680, after it goes to the photosystem 1, it gets
Yes.
reduced.
P680 gets oxidized. So when it loses those electrons, it's going to get oxidized, right?
Yeah, but then when it reacts with water, it gets reduced.
Right, correct. That's where water comes in. That's why we need water, is so that we can get those electrons back into photosystem 2 to reduce it.
All right, thank you.
Yeah. Okay, so next we'll talk about the dark reactions. So dark reactions are going to occur in the stroma. And like I said before, it's going to use the products from the light reaction, NADPH and ATP. But it doesn't need the light energy. So it doesn't need the photons to strike anywhere in the dark reactions. So it uses the products from the light reaction. So it's not totally light independent, but it doesn't need that light energy. So there are 3 main stages. So the first stage is carboxylation, second stage is reduction, and the last step is regeneration. In these reactions, we're going to use an enzyme known as Rubisco. That's important for carboxylation, and we'll get into these after I explain this diagram and what happens. So first, we're going to start off with ribulose 1,5-bisphosphate, which is a 5-carbon molecule. And we're going to start off with carbon dioxide as well. This reaction is going to become a carboxylation reaction. And what that means is basically we're going to add that carbon from carbon dioxide into the ribulose 1,5-bisphosphate, and we're going to make a 6-carbon molecule. And this process right here happens with the help of the enzyme Rubisco. Okay, so once we have our 6-carbon molecule, it's going to get split into a 3-carbon molecule, which is known as 3-PGA. So since it's a 6-carbon molecule, we're going to get 2 of these. So when you add that together, it's going to come out to 6, right? So that's what the carboxylation part of cellular respiration does. Does that make sense? Any questions on that? Okay, so we'll go on to the next step, which is reduction. In reduction, what's going to happen is we're going to use ATP and NADPH to essentially reduce this molecule right here, 3-phosphoglycerate. When we reduce that molecule, we're going to make a triose phosphate. So as I said before, we're going to have 2 of the 3 PGA molecules. So for one PGA molecule, we're going to need 1 ATP. So if we have 2 PGA, we're going to ultimately need 2 ATP for that. And same thing with NADPH. So 1 PGA will require 1 NADPH, and 2 PGA will require 2 NADPH. So, and we can say that for every carbon dioxide molecule that enters the Calvin cycle, so for every carbon dioxide, we're going to need 2 ATP and 2 NADPH for the reduction. So just for the reduction stage, for every molecule of carbon dioxide that enters the Calvin cycle, we're going to need 2 ATP and 2 NADPH. Okay, and what that's going to ultimately produce is 2 triose phosphates, or also known as G3P. Okay.
Is this the same as PGAL?
Yeah, I believe it's the same thing.
Okay, thank you.
Yeah. So next, we're going to go into regeneration. And in regeneration, the G3P molecules that we get from the reduction phase, only a few of those are going to be used. Actually, only one of that is going to be used to make glucose. And the rest are going to go into regeneration of this molecule known as ribulose 1,5-bisphosphate. And that happens just because we need the cycle to continue. And if we look at this diagram here, if we start off with 3 carbon dioxide, we're going to ultimately make 6 G3P molecules. And one of that is going to go into making glucose. And the other 5 is going to go into regeneration. So if we think of this, if we think of this in terms of like how many cycles are we going to need to make a glucose molecule? Well, this right here represents, this diagram is going to represent 3 cycles of the Calvin cycle, right? Because we start off with 3 carbon dioxide. And if 3 cycles will give us 1 G3P that goes into making glucose, how many cycles are we going to ultimately need to make 1 molecule of glucose, considering that glucose is a 6 carbon molecule? Can anyone answer that? Just guess.
Would it be 18?
No.
Would it be 6?
It would be 6. That's right. So, like I said, so for three cycles, we're going to make 6 G3P molecules. One of those is going to go into making glucose, and the other 5 is going to go into regeneration. Since glucose is a 6 carbon molecule, right, we only have 3 carbons here, so we can't make glucose with just 3 cycles of the Calvin cycle. We need another additional 3 cycles. So when we have a total of 6 cycles, we're going to produce another 6 G3P, and then it's kind of the same process. The 5 will go into regeneration, and the 1 will go into making glucose. So when we have 6 cycles, we're going to ultimately have 2 G3P molecules. Does that make sense? Any questions on that? Okay. If you have questions later, just let me know. All right, so we'll do a little bit of practice with this, and this was kind of just the turning point clicker questions that we did in class. So mass of a seed is 1.5 grams. What effect would these conditions have on the effect of the dry mass of the plant? So no light and no water. What would happen there? Would it be less than, greater than, or equal to 1.5 grams?
Wouldn't it be
Equal
equal to?
to?
Yeah, so it'd be equal to. You guys know why that is?
It doesn't germinate and you don't have any water to like.
Right. So we don't have light, so that means we don't have photosynthesis. We don't have water, so that means we can't do any cellular respiration. So the mass of the seed is not going to change at all. Okay. So for light and water, what would happen? Would it be greater than, equal to, or the same?
Greater than.
Okay. So why is that?
Because we have both light and water in order for photosynthesis to occur.
Right. So the plants are going to be making the ATP. They're going to be growing, and it's going to build up that the mass of the plant essentially. And for no light, and if we have no light but we have water, what would happen there?
Would it still be greater than, I believe?
No.
It'd be less than.
Less.
So it's going to be less than. So does anyone want to explain why that is?
Aren't you releasing? So you don't have light, but you can do it with the Calvin cycle. So once you do it with the Calvin cycle, aren't you releasing? Sorry, wait, hold on. So aren't you releasing carbon dioxide?
You are releasing carbon dioxide, but not from the Calvin cycle.
No, sorry, that's from. Sorry, I just lost it.
That right?
What was that?
The carbon dioxide, it comes from the plant, not from the cycle.
What?
I'm confused with that question.
Plant is doing the process.
Okay, I'll explain why. So if we have water, our dry mass for a seed is 1.5 grams, and that mass, where it comes from, is if we have a seed, it's going to contain like a lot of starch, which we can use for cellular respiration. And it needs that water to start cellular respiration. And in cellular respiration, what we have is a glucose molecule that's going to be going in, that's going to be breaking down and making ATP and releasing carbon dioxide, right, into the atmosphere. So
That's in glycolysis, right? Sorry. Like releasing the carbon dioxide is in glycolysis.
Releasing carbon dioxide is in the citric acid cycle.
Okay, all right, thank you.
Yeah. So since we're doing cellular respiration on that with water, it's going to be losing all that mass that it had, that dry mass from starch. So we don't have that mass anymore, and it's going to be we're going to use it to make kind of energy for the plant. But ultimately, what I said, or ultimately what happens is when you dry the plants and the water kind of evaporates, the mass is going to be less than that of a seed. Okay. So this question here asks, what part of the photosynthetic pathway is defected, if any, for this plant? So we have our chlorophyll absorption to be the same. oxygen production is the same. And this is in comparison to the wild type. So equals mean wild type. Our NADPH production is less than wild type. ATP synthase activity is the same. And our G3P production is less than wild type. So what part of the photosynthetic pathway would be defected? Do you guys know?
The citric acid cycle?
The last one, whatever we just went through.
So for photosynthesis, not cellular respiration.
It's the one with like the carbon fixation reduction, and then
That's Calvin.
the the Calvin cycle.
The Calvin cycle?
Yeah.
Okay, you're right. The Calvin cycle is going to be affected by it because we're going to have less G3P production, right? But ultimately, we have less NADPH production as well. And where do we get the NADPH that we need for the Calvin cycle?
The electron transport chain.
So what part specifically of the electron transport chain for photosynthesis?
Photosystem 1.
Photosystem 1. So this happens at photosystem 1, right? So if we have a decrease in NADPH production, we know that there's something wrong with photosystem 1.
But in G3P production, in the Calvin cycle, you're also making NADH, which is used in the electron transport chain also. So can you use that reasoning as well? Because the Calvin cycle is replenishing the NAD plus molecules for the light-dependent cycle.
So for the light-dependent cycle, we're going to end up getting NADPH and ATP. Those NADPH are going to be used here in the the Calvin cycle right here. So this is where the NADPH is used.
Right, but it's being produced, the NADP plus, it's being produced in the dark reactions. So I'm saying if there's none in the dark reactions, then you will have less NADPH in the electron transport chain as well.
Yeah, so yeah, that's what happens. We don't have the NADPH in the electron transport chain, enough of it. So that's why there's a defect in our photosystem 1, right? So up here, we said that photosystem 1 is right here. It produces our NADPH. And if we don't have enough of that NADPH, we know that there's something wrong with photosystem 1. If you kind of inhibit photosystem 1, just take it out,
right,
we won't get this NADPH production, right? We're not going to get that. So if there's a decrease in NADPH production, that means that there's something wrong with photosystem 1, because that's where we get our NADPH from for photosynthesis.
Right, but if you don't have any NADP plus from the Calvin cycle, you're not going to have any for the light-dependent reaction. That's what I'm saying.
The NADH has to come first.
Yeah, we have to get the NADPH from the light.
Because, like, for all we know, they could just be making a whole lot of NADP, but then it's not coming through. So unless they specifically say there's a decrease in NADP, you can't, like, for sure say that there's a problem with the production.
Yeah. Also, so yeah, this is talking about the photosynthetic pathway. You don't need to think about cellular respiration here. Just think about what's happening in photosynthesis, where we get that NADPH in photosynthesis, and what part of photosynthesis makes the NADPH. And if you can know that, and if you know where it makes the NADPH, which is photosystem 1, then you know kind of what's defected, right?
Thank you.
Yeah. Okay. So that's all I have for photosynthesis. I know that the Calvin cycle is a little confusing, and I wrote a lot here, and it probably may not have made sense to most of you guys. So if you guys have any questions over that, I can do my best try and explain it again and go a little bit slower. But Dequan and I will just be answering questions right now. So
somebody wanted to cover anaerobic respiration.
anaerobic respiration?
Yeah, it was let me see. Oh, it was V. V, do you want to cover the like when with anaerobic respiration, do you want to go over like fermentation pathways or just kind of how the ATP is reduced, why that is?
So for the exam, you don't need to know anaerobic respiration. We can go over that if you want.
Oh no, if we don't need to know that, I'm good. Thank you.
I posted a bunch of links for you guys, like over cellular energetics, photosystem, or photosynthesis, and metabolism in regards to anabolism and catabolism. Definitely check those out. Those will help. And there's so much resources you guys can use, especially with Khan Academy, and it's all free. Yeah, so definitely get after it, get a good grade on the test. If you guys need anything else, we just hang out. I'll probably just hang out till everybody's off, and then I'll get off.
Thank you.
Yeah, you're welcome. Jay, what do you use to make your things? That looked really good.
I just used the diagrams, you mean?
Yeah, are those like screenshots that you inserted into some sort of?
Yeah. So it's just one.
Oh, okay. So you're using OneNote with screenshots. Cool.
Yeah, just kind of copy and pasted these diagrams on there.
I'll try to post the recording sometime this evening. Dr. Bang was saying it takes a long time to upload it, like so long that he would start uploading it and it wouldn't upload till the next day. So we'll see. It might have something to do with the Wi-Fi. You'll know it's up. I'll send out an announcement. Just letting you all know that it's posted.
Yeah, thank you guys. This has been really helpful.
Yeah, for sure. You're welcome.
Can someone explain when a seed is germinating? When does it start photosynthesis?
It starts photosynthesis when it can access the light. So when it's germinating, it's going to be under the soil. It's not going to have any access to like light of any type. So during that part of germination, all the seed is going to be trying to do is make energy for it to grow and kind of sprout out of that. And the energy that it gets is from cellular respiration, because a seed has like stored energy in the form of starch, and it uses that starch through the cellular respiration pathway once it germinates, and it has the access to the light. Then that's when it starts photosynthesis.
All right, thank you so much.
Oh, no problem.
Cool. That's everybody.
Everyone's gone.
Yeah. So I'm going to stop